First, I would like to thank the organizers for giving me this opportunity to present our work. And before I start, I just want to say that this project was done in collaboration with Avi and Sichi, who are two other graduate students in our lab. It's been a really fun collaboration, and so I'm really excited to tell you about what we've done today. So the phenomenon that we're interested in and that really got us talking to each other is the fact that gene expression is noisy. So when you take a reporter gene and you integrate it into the genome, for example, here at GFP, and you generate a clonal population of cells that all have GFP integrated in the same location, you get this kind of expression pattern where some cells will have more GFP expressed and some cells will have less GFP expressed. And now if you draw a distribution where GFP expression is on the x-axis and on the y-axis is probability, you would get a distribution that looks something like this. And when we really measure reported gene expression in bulk, what we're getting is the mean of this distribution. But each gene also has its associated variance, and this variance is what we're really interested in studying. So when I say variance or noise in this talk, I'm really referring to the same thing, and I'll be using these terms kind of interchangeably. There are a lot of reasons why the variance is important, but one of the oldest examples that we know of is position effect variegation, or PEV. And the classical example really is if you take a gene that encodes for the red protein in a fly's eye and you integrate it in a euchromatic locus, you will get the red gene being expressed in all the cells in the eye, and you get a phenotype that looks something like this. However, if you now take the gene and you move it close to heterochromatin, you get heterochromatin spreading into the gene, which will turn off the gene in some cells, so you'll get white cells, and you'll leave the gene on in other cells, and you'll get red cells, and you end up with a variegated phenotype like this eye here. Now you can imagine that the distribution of red protein in these cells will look quite different. So if you have this dotted line, which is a theoretical kind of boundary where everything on the right is going to be a red cell and everything on the left is going to be a white cell, you can imagine that the distribution for a euchromatic locus would be more like this, where it's mostly on the right, while the variegated eye will have a lot more on the left of this line. So the question that we're interested in asking then is, what are the sources of this gene expression noise? And PEV really gives us some clue. These classical studies have given us some clue that it has something to do with chromatin modifiers. Right? So if you take this variegated eye and you look for suppressors of variegation, so things that reduce heterochromatin spreading, the red gene is now expressed in almost every cell. Or you look for enhancers of variegation where there's much more heterochromatin spreading and the gene is basically silenced in almost every cell. You'll find that a lot of these suppressors and enhancers of variegation are actually going to be chromatin modifiers, which told us that maybe chromatin environment is something important for regulating gene expression noise. So then our specific question really is, how does the chromatin environment affect gene expression noise? And to answer this question, our goal was to develop a technology for measuring the expression noise of the same integrated reporter in many genomic locations in human cells, with the idea that different genomic locations will have different chromatin environments that we can then correlate with expression noise. So to do this, we're actually using a method that already exists. It's called TRIP, stands for Thousands of Reporters Integrated in Parallel, and was actually also developed in the von Steenzel lab. And what TRIP allows us to do is measure the mean expression of reporter genes at thousands of locations in parallel. So TRIP starts with a library of reporters with random barcodes. Each of these reporters has a CMV promoter driving the expression of a BFP reporter gene and a barcode in the 3' UTR. So we clone a library of hundreds and thousands of these barcodes, and each reporter is cloned between these inverted terminal repeats that allow us to integrate the library into random locations with piggyback transposase. Now you can imagine that a gene that's integrated into a more open chromatin region will probably give you more mRNAs, while a gene that gets integrated somewhere more closed, more repressed, will give you less mRNAs. And because each integration is uniquely barcoded, what we can do is count the DNA and mRNA barcodes by sequencing. We can also map the locations of the integrations by associating the barcodes with its genomic region. And so we can really get the mean expression of each barcode, and we know at each genomic location. What TRIP allows us to do then is answer the question, how does chromatin environment affect expression mean? So here on the x-axis, I'm showing you the ChromaHMM segue combined annotation, which is an annotation based on the chromatin environment. So it takes a lot of different chromatin chip-seq data from K562 cells, which is the cell type we're working in, and comes up with these six or seven different annotations. And on the y-axis here, I'm showing you expression mean. And so each violin plot really represents the expression of reporters that have been integrated into each of these different kinds of chromatin. And what you can see is basically what you would kind of expect, where more active chromatin regions, such as transcribed regions or enhancer regions, have higher mean expression levels compared to repressed regions or CTCF regions. But what we don't know is how this plot is going to look like for noise. And so what we aim to do is kind of fill in this blank over here. So the method that we're using is derived from TRIP, and we're calling it single-cell TRIP or scTRIP. And it starts pretty much in the same way as regular TRIP with a library of randomly barcoded reporter genes. One difference is we added this character sequence here in the 3' UTR, and I will explain why in a bit. Now, when we integrate the library into random locations with piggyback transposase, what we're really interested in is looking at its expression in single cells. This is a single-cell biology conference after all. And so you can imagine in these 3 cells, they have the same integrations and the same barcodes. But for example, this yellow integration here, it might produce 3 mRNAs in one cell, 1 in the other cell, and the same for all the other mRNAs. So what we're interested in doing is counting the number of mRNAs per cell by 10x sequencing, and again, also mapping the locations of the integrations so we know where they are. So how do we do this by 10x sequencing? Each 10x gel bead actually comes with 2 different kinds of sequences. The poly-T sequence captures the poly-A mRNAs, but the capture sequence was actually designed for perturb-seq applications so you can capture the guide RNAs in the cells. But I told you earlier we included the capture sequence in our 3' UTR, so we can capture the poly-A's, our mRNAs by poly-A, as well as by capture sequence. The idea being that the poly-T sequences will be busy binding to all the other poly-A mRNAs in the cell, while the capture sequence will actually be free to capture just our reporter gene. Now we can design primers to specifically amplify our reporter gene from the 10x pool. So when we sequence, we're really only sequencing the barcodes from our reporter genes, and we're not sequencing the entire transcriptome. And that really just saves us a bunch of sequencing costs. So when all this is said and done, what we get is this table of UMI counts, where each column is a different cell, each row is a different location or a different barcode, and we can count the number of UMIs to tell us the number of mRNAs, basically like regular single-cell RNA-seq. Using this table, we can generate a distribution for each location. So location 1 might look something like this. Location 3 that maybe has more RNAs will look something more like this. And using these, we can calculate a mean and a variance for each location in the cell. So now I'm going to show you the first piece of data that we have, which is basically that single-cell TRIP measurements are reproducible. So on the x-axis here, I'm showing you replicate 1. On the y-axis is replicate 2. And each dot really represents the mean expression of a given location. And you can see that they're pretty correlated over about 930 locations that we've managed to measure. Now, if we look at variance, which is what we're interested in, so now each dot represents the variance of the location, you can see that they're also pretty correlated. So we're fairly confident in these measurements. We also validated these measurements by single molecule FISH. So we generated 2 clonal cell lines. They have an integration of the reporter gene in a different location. And we perform single molecule FISH on each of these 2 clones. So actually, scTRIP performs single molecule FISH. And then we count the number of mRNAs in the cell. And what we can see is that when we compare single-cell TRIP and single molecule FISH, the means of location 2 is always higher than location 1. And so the same for noise. And we can validate this with single molecule FISH. So this suggests that our measurements are indeed pretty good. So finally, we can answer the question, how do the chromatin annotations really affect gene expression noise? So here on the x-axis, again, I'm showing you the chromatin segway combined annotations. And on the y-axis now is noise. And in general, across annotations, it does seem like there's not much effect on noise. Repressed regions, though, tend to have slightly higher expression noise than the other regions. And this kind of tells us that maybe different kinds of chromatin environments can have a different effect on gene expression noise. We've actually also validated this by single molecule FISH. Here I'm showing you about 20 clonal cell lines. About half have an integration in a transcribed region and about half have an integration in a repressed region. And in the repressed regions, there tends to be more noise than transcribed regions too. So finally, what I've shown you today is that single-cell TRIP is a technology for reliably measuring noise across the genome, and that different chromatin environments can have different effects on noise. And so I'd like to thank my lab just for being an incredibly supportive environment, and again, Avi and scTRIP for working on this together with me. And I'll be happy to take any questions.
Thank you, Clarice, for a lovely talk. That was really very clever technique. So, Aaron, do you have a question? I think you're on mute.
Muted. Really nice talk, Clarice. I was wondering if integration efficiency is variable based on the heterochromatin environment. So like if you're sort of only seeing certain areas.
So yes, there's definitely going to be a bias for active chromatin regions compared to heterochromatin regions. But because we do see integrations in repressed and as well as heterochromatin when we use kind of other chromatin annotation type of things, we think that we are kind of capturing these regions, maybe not as high numbers as the rest, but we do capture it, and we're working on scaling this up so we get more of these repressed regions as well.
Thank you.
Cool. I had a question. So, I mean, there was some sort of modest effect of noise in repressed regions, just if you look at sort of the median change.
Yeah.
But what was interesting to me is there are a lot more outliers in the repressed region. I guess what I'm wondering is, rather than say, oh, well, these are the categories of regions and here are the noise profiles of various integrations in those regions. But if you kind of flipped it around and said, okay, well, let me take all the high noise integrations and then ask, like, what is common about those?
Yeah, so that's interesting. That's something we haven't done yet. What's really interesting is actually we've done this where we've kind of sampled, like if you sample these locations and just simulate like small numbers of locations, you will never see a difference, even whether in mean or in noise levels. You have to kind of sample like large numbers of regions before you start seeing differences in mean and noise. And we think it's because there really are a few outliers that really drive the differences in mean and noise in these regions. So it's definitely something that we're interested in like furthering, but this is still pre-early stages. So.
Cool. Okay. We got a couple audience questions here. So Sanjay Srivatsan asks, what proportion of the variance is left unexplained after accounting for those genomic labels? Are there any plans to sequence the RNA to get a rough sense for other features like cell cycle?
So I'm not sure how much is left unexplained. We didn't quantify how much the chromatin labels explain. We definitely corrected for mean expression levels when we did noise measurements. So I can't answer that question now. But as for cell cycle, we've actually looked at single molecule fish, and we have found no correlation between cell cycle and our results in single molecule fish. So we don't think that cell cycle is playing a huge role in the variance measurements.
Yeah. So Shweta Murali asks, do you find the overall distribution stays the same with an offset for repressed versus not? Or does the distribution itself change as well?
That is a good question. I think most of the distributions look fairly similar. They are not that different across the different locations, but we definitely haven't looked very closely yet. And yeah, but I think they look mostly the same, if I'm not wrong.
Yeah, it's kind of, it's an interesting question. I mean, I feel like, I guess, so one question I have is how much is the distribution dependent on the particular promoter that you put in? So if you had put in some other type of promoter, would you get, one of these like tail type distributions?
So this is a whole other paper that actually Suchi is working on right now with single molecule fish, where he has actually integrated different promoters in the same genomic location. And actually what he finds is I think promoters don't really affect, the effect is independent of the location. So whatever effect the promoter has doesn't matter where it's integrated. But for the exact details, you can talk to Suchi, who's also at this conference. He'll be very interested to talk to you about this.
Cool, yeah. Veronique LeBlanc asks, is there a relationship between expression mean and noise?
Definitely. So mean and noise are definitely correlated, but we definitely see regions where they have the same mean and like almost tenfold difference in noise. So mostly, I think mean can explain maybe 60 to 70% of the variance, but there is some effect on mean independent variance as well.
Cool. And we've got one last question here. So Joshua Thyssen asks, how many integration events occur per cell line? Does relative expression noise across integration sites show global patterns in one cell relative to other cells?
So we get about 20 integrations per cell, and we haven't been able to we haven't linked the barcodes that are in the same cell. So because we are pooling cells, different cells that each have 20 integrations, we haven't really figured out which integrations are coming from the same initial clone yet. So we can't answer that question, but we're definitely looking forward to doing that. But we get about 20 integrations per cell. So, yeah.
Perfect. Well, thank you very much, Clarice.